We all know that the cell is the basic unit of life. But why is it called the basic unit, and how can something so tiny control life itself? In this upcoming series, we'll explore that in depth. So, let's begin with a simple question. What are cells? Cells are microscopic structures that form the foundation of all living organisms. They carry out every activity necessary for life, from producing energy to growing, repairing, and reproducing. When we examine the levels of biological organization, the cell is the level where life truly begins. In unicellular organisms, a single cell performs all life processes on its own. But in multicellular organisms like plants and animals, millions to trillions of cells work together in coordination to sustain life. Just as bricks build the walls of a building, cells build complex living organisms. From the simplest bacterium to the largest animal on Earth, and from microscopic algae to towering trees, every living organism is made of cells. When we say a cell carries out all activities of life, we mean that nature has placed an entire system inside something unimaginably small. Think of a cell as a city. A city has a power plant that generates energy. A cell has mitochondria that produce energy. A city has complex road networks and transportation systems. A cell has intricate pathways that transport materials. A city has a control center that manages operations. A cell has a nucleus that acts as the control unit. All of these complex systems exist within a space too small to see with the naked eye. That is not ordinary. That is precision. That is design at its finest. And this is just the beginning. In the next part, we will explore how scientists discovered cells and how this discovery changed biology forever. But if cells are so tiny that we cannot see them with the naked eye, an important question arises. How did scientists discover them in the first place? The discovery of cells became possible only after the invention of one important scientific tool, the microscope. In 1665, Robert Hooke, an English scientist, examined a thin slice of cork under a microscope. What he saw surprised him. Instead of a solid structure, the cork appeared to contain many tiny box-like compartments arranged side by side. These compartments reminded him of the small rooms used by monks in monasteries. Because of this resemblance, Hooke called them cells. However, the structures he observed were actually dead plant cells. Hooke had only seen the empty walls of cells, not the living contents inside them. Still, his observation introduced the world to the concept of the cell. A few years later, Anton van Leeuwenhoek, a Dutch scientist, built powerful microscopes using carefully polished lenses. When he observed water samples, plaque from teeth, and other materials, he saw something extraordinary. He observed tiny living organisms moving under the microscope. These organisms were far too small to be seen by the naked eye. Leeuwenhoek called them animalcules, meaning tiny animals. Today we know them as microorganisms. This was the first time scientists had ever seen living cells. These early discoveries opened the door to a completely new understanding of living organisms. Scientists began to realize that life was not made of solid material, but instead was built from countless microscopic units working together. But this raised an even bigger question. Are all living organisms made of cells? The answer to that question eventually led to one of the most important principles in biology, the cell theory. Over the next 2 centuries, scientists carefully studied plants and animals to find the answer. Their observations led to the development of one of the most important ideas in biology, the cell theory. In 1838, Matthias Schleiden, a German scientist, studied plant tissues under the microscope. After examining many different plant samples, he concluded that all plants are made of cells. In 1839, Theodor Schwann, a year later, another scientist, studied animal tissues and discovered that animals are also made of cells. This was a groundbreaking realization. It meant that both plants and animals, despite their differences, shared the same fundamental unit of structure, the cell. In 1855, Rudolf Virchow, a scientist, added another important idea. He stated that all cells arise from pre-existing cells. In simple terms, this means that new cells are formed only from already existing cells. Together, these discoveries formed what we now call the cell theory. Cell theory can be summarized into three fundamental statements. All living organisms are made up of one or more cells. Some organisms like bacteria consist of just a single cell. Others like plants and animals are made of millions or even trillions of cells. The cell is the basic structural and functional unit of life. This means that the cell is the smallest unit capable of carrying out all the essential processes of life. All cells arise from pre-existing cells. Cells do not appear spontaneously. They are produced when existing cells divide. Cell theory changed the way scientists understood life. It showed that despite the enormous diversity of living organisms from microscopic bacteria to giant trees and animals, life follows a common biological principle. Every living thing begins with a cell. From that single unit, life grows, develops, and becomes the complex organisms we see around us. But if all living things are made of cells, another important question arises. Are all cells the same? The answer is no. Based on their structure and level of complexity, cells are classified into two major categories. First, prokaryotic cells, and second, eukaryotic cells. This classification helps scientists understand how different organisms are organized at the cellular level. Prokaryotic cells are the simplest and most primitive type of cells. These cells do not have a true nucleus. Instead of being enclosed within a nuclear membrane, the genetic material is located in a region called the nucleoid. Prokaryotic cells also lack membrane-bound organelles such as mitochondria or Golgi bodies. Despite their simple structure, these cells are capable of carrying out all the basic activities necessary for life. Examples of organisms with prokaryotic cells include bacteria and archaea. These organisms are usually unicellular, meaning the entire organism consists of just one cell. Yet even a single prokaryotic cell can grow, reproduce, obtain energy, and respond to its environment. Eukaryotic cells are larger and more complex. Unlike prokaryotic cells, they contain a true nucleus surrounded by a nuclear membrane. This nucleus stores the genetic material and controls the activities of the cell. Eukaryotic cells also contain membrane-bound organelles, each performing specific functions inside the cell. Examples include mitochondria for energy production, endoplasmic reticulum for synthesis and transport, Golgi apparatus for packaging and distribution of materials. Eukaryotic cells are found in plants, animals, fungi, and protists. Many of these organisms are multicellular, where specialized cells work together to form tissues and organs. In simple terms, prokaryotic cells are small, simple, and lack a true nucleus. Eukaryotic cells are larger, more complex, and contain a well-defined nucleus along with specialized organelles. This difference represents one of the most fundamental divisions in the living world. Understanding these two types of cells helps us see how life ranges from simple microscopic organisms to highly complex living systems. But now another question appears. If eukaryotic cells contain many internal structures working together, what exactly are these structures and what do they do? In the next part, we will explore the structure of the cell and the different organelles that keep the cell functioning like a perfectly organized system.